Okay, well, it's a pleasure to introduce our speaker today, Wayne Getz, who is in ESPM and who has been at the University of California at Berkeley since 1979, so he has a long history here. He tells me he last spoke at MVZ Lunch about 5 years ago. We had a wonderful chat this fall, and I said, you've got to come talk to the MVZ. It would be wonderful to hear about what you're doing. Wayne was trained as a mathematician in South Africa and has a long history of studying disease ecology and epidemiology, and had a paper published in 2005 in Nature that's been very influential on super spreading in disease, a paper modeling variation in among individuals and the spread of diseases, and that's been cited nearly 400 times, so just in the last few years. So today he's going to speak to us about simulating evolutionary processes in complex environments. So thank you very much, Wayne, for coming.
Well, thanks, Michael, and it's great to be here. And I'm always very envious when I come in here because there's a sense of a community. And sometimes the rest of us sort of drift outside on other labs and don't quite have the big community that you have here, which is really terrific. So I have an obnoxious title, a computational toolbox for dummies. By that I only mean coding dummies, and by dummies I include myself. So the person who's not a dummy is Richard Salter from Oberlin College, who's really the computer scientist and the architect of this platform. And I just kind of egg him along and say, we need this, we need that, we need to be able to do all these different things. And then miraculously, he comes up with ways to do all these things. So what I've been on about is that I'm very interested in multi-scale modeling, and particularly what I call linking the inner and outer world of organisms. And so I'll go into that in a little more detail in my talk without dwelling on it too much at this point in time. And what we really need at this stage is a cultural revolution in what I call computational population biology. And I'll explain a little what I mean by that, but this is where we are. You know, so you see the orbit of a planet is elliptical. What's an orbit? What's a planet? What's elliptical? So, you know, we need to be able to come together to communicate better. And hopefully that this talk that I'm going to give will be part of this communication process. So as an outline of this talk, what I'm going to do is I'm going to describe some past work in what I call computational population biology that was implemented using MATLAB. And again, I didn't write the code. It was done by a student from Sweden who worked with a colleague of mine from Sweden and spent a lot of time here during his PhD years. Then I want to describe what I mean by the emerging field of computational population biology. Then I want to go on to describe the characteristics of a good computational platform as embodied in the architecture of Nova. And I think Nova at this time has some outstanding features that should make it very attractive. It's a work in progress and development, but in the last 2 years we've come quite a long way. And then I want to apply Nova to the construction of genetic algorithms to investigate problems in movement and resource ecology. I'll set up the idea of genetic algorithms in the early work that I've done that was done through coding up the problem in MATLAB. So past work in computational biology using MATLAB. This work is based on modeling or agent-based modeling. So there are two modeling paradigms that are very prevalent. The one is using systems of ordinary differential equations or difference equations. Sometimes people describe spatial processes in terms of partial differential equations, but when you discretize it, it also comes down to systems of ordinary equations or ordinary differential equations. And we use neural networks, and what that really means will become evident in the talk. And one of the things I'm particularly interested in is consumer resource processes. And so we're interested in how consumers and resources interact. And this work focuses on some of the co-evolutionary aspects of this process to address a bunch of questions. And this work was started 15 years ago, and most recently we published something in PLOS One, which I guess is after you've banged your head against 3 or 4 other journals who say, I don't want your stuff. We take it to PLOS One. We think this is very good stuff, but obviously it's quite complicated, and so it doesn't fit into any great existing paradigm. So PLOS One is a good outlet for that sort of work. So we started out with the following biological question, which I became interested in. I don't know how many of you know Liz Bernays and Reg Chapman, the late Reg Chapman. The older faculty certainly do. And I used to be in the division of biological control and talked to them a lot, and became interested in the question of why are there so many insect herbivores in heterogeneous landscapes where you have a lot of food that they could eat, but they're not eating. So why do they specialize by choice? A lot of Liz's work was to show, and Reg's work to show, that grasshoppers, for example, can be force-fed stuff that they don't eat in nature, and they thrive on it. So this is a bit of a puzzle. So as part of this biological question, we thought, well, how does the complexity of the plant, what I call informational landscape, where you might have odors, colors, shape out there that the animal can perceive, affect the ecological structure and evolutionary history of insect herbivore guilds? So how do these guilds get set up? What does the degree of specialization look like? How does it change over time? So another question relates to the evolution of Batesian mimicry, where you have a palatable species that's unprotected trying to look like a non-palatable species that's being ignored by the primary predators out there. So how does the evolution of Batesian mimicry affect the guild structure of the exploiters? And finally, how does this all, the big questions relate to, how does speciation take place, particularly sympatric speciation? We all know how geographical isolation can lead to speciation, but this is an interesting question. And of course, many of you might be familiar with the Ragalitas story, which is maggot flies on hawthorn and apple, and studies by Guy Bush et al. In the 70s showing that the gene flow is reduced between the two types that exploit the different resources. So being a mathematical modeler, not being a field biologist, I try to understand things in terms of building models, which is, I guess, an easier but maybe more boring lifestyle. But I do get to travel a lot to field sites, so I get students who are field biologists, and then I have them hot and sweaty collecting data, and then I go visit them for short periods of time, and I don't suffer a lot. So one of the things I got interested in was olfactory processing, and we applied this to this particular model. This provides kind of the leitmotif of the model. Think of a plant putting out a signature, a chemical signature, being picked up by an insect. This is a picture of its brain. This is actually an artificial neural network in which the signal comes in to this processing. This is in the antennal lobe of an insect, and then this information is summed up and goes through a transducer of sorts, and then goes to a second layer and a third layer and maybe many layers, and out comes an output that says 1 or 0, exploit this plant, don't exploit this plant, etc. So that's our basic model. The red's not showing up very well, but the model components, firstly in asking questions about the evolution of specialization, we used agent-based models of evolving exploiter decision-making traits. So each of our individuals was a little neural network in a computer, and it could evolve. And it evolves in response to the signal that's coming in, and it looks at the signal, and the evolution says, use this or don't use this. So ahead of time, we don't know whether to tell it to use it or not, and so we use genetic algorithms to address this problem. So the agents have to be replicated with the decision-making traits intact with some mutation. So if we've got a little neural network and it performs well in that it collects a lot of resources on a landscape, then we take those that collect the most resources and we say, you've got a good set of weights in your little processor, so we're going to replicate you, but we'll make some perturbations to that. And we also have a fitness function that is influenced both by competition and the quality of victims. So for example, in our plant space, there's some plants that may be protected. That's a poor quality victim, or there may be plants that are not protected by, say, chemical compounds on which the individuals can thrive. That's a good quality plant. But if there are a lot of individuals exploiting the same plants, then there's going to be interest-specific competition. So we set up these fitness functions as well. So we have this ecological background to these agents moving in this ecological space. And we make this space heterogeneous in the sense that animals are going to encounter plants of different types, where the types are based on the signal that they put out. So the animal reads the signal and says, oh, you're a plant of type 1. You're a plant of type 2, or if it hasn't evolved to the point where it can discriminate between these, it just says, oh, you're a plant and I'll use you. So what it really has to do is it has to learn how to discriminate between the good plants and the bad plants in terms of those that are protected using some kind of, whether it's physical structures or chemical compounds. And the fitness of the exploiters increases with the quality of the resources they use, and the fitness of the exploiters also decreases depending on the intensity of competition they encounter on these different plant types. So as part of this study, in the second part, we looked at the evolution of mimicry, where we said plants that are vulnerable might try to put out a signal that looks like plants that are not vulnerable. So for example, if an exploiter has evolved to exploit plants that are vulnerable and not use the plants that are protected, then the plants that are protected try to look more like the plants that are vulnerable to escape this discrimination that the exploiters can set up. Now I'm using exploiter, victim, but you can think of insect, plant if you want. So insects are the exploiters, plants are the victims in this case. And so the relative fitness of the plant types relates to the intensity of the exploitation upon them. And so high quality plants can reduce that intensity by trying to mimic the signals of low quality plants that the exploiters have evolved to avoid. So that's the co-evolutionary part, or the evolution of mimicry under co-evolutionary situation. And then we're interested in some speciation. And to address this question, our final study, which we published in PLOS Biology, takes parthenogenetic reproduction or cloning and puts sexual reproduction in place of it, and also adds some genes for mate selection, where the genes relate to the fact that individuals are more likely to mate with individuals exploiting the same plants as them as exploiting different plants. So that sort of fits in with the kind of Ragalitas, hawthorn, apple situation. And so we include genes for both perceptual characteristics. Whenever you make a mathematical model, you have parameters, and these parameters have values. And in a computational model, these values can be thought of as reflecting something in the genetic underpinnings. And so when you mutate your individuals, what you're doing is you're slightly changing the values of the parameters that you work with in the model. And so finally, after we analyzed all this, we said, can we identify some interesting mechanisms that are causing the reduced fitness of hybrids? Because the reduced fitness of hybrids is what keeps these two populations apart if they're incipient sympatric speciation. So the exploiter phenotypes, so each agent in this case has what we call a three-layered perceptron. So the signals come in, these signals get added over here, they get put through a kind of a step function, or a gentle step function you can think of as kind of a logistic function or a ramp function. And so this is called a hidden layer, and there are a number of nodes in this hidden layer. So we have two inputs, we can have 3, 4, 5, 6, depending on the discrimination task. And then these come out, they're added in here, and they go through. And once you do the addition over here, the addition might land up on this part of the curve, in which case it's a 0, or this part of a curve, in which case it's a 1, or something intermediate, which is a small part of the region. But generally you're getting numbers out of here that look like zeros and ones, and they're recombining here, and we go through the same process until finally you come out with a 0 or a 1. If it's a 0, it means this signal that's coming in, avoid it. And if it's a 1, it means this signal coming in, use it. So this is kind of what we call a classifier. So it's a machine that does classifications. And we could have a high-dimensional input space. And if we want to do more than yes-no classifications, if you've got n outputs, then you can get 2 to the n different categories. So this is an artificial neural network, also known as a multi-layered perceptron, that does classification. And these individuals are flying around, they're looking at the inputs, and they're classifying them into, this is something I want to exploit, or this is something I don't want to exploit. So basically the input is the victim signal, the output is don't lay an egg, or lay an egg in the context of, say, flies exploiting hawthorn versus apples. And the decision-making phenotype depends on the weights that we don't see here, that you take the signal and you take a weighted sum, you take the signals, you take a weighted sum. Those numbers are the things that we play around with. Those are the parameters, and those are the things that we change in a genetic algorithm. And so the phenotypes, those individuals that collect the most resources, we replicate them, we give them the same parameter values plus some mutation. And the probability for an egg to hatch and produce an adult depends on the relative number of eggs laid in that habitat, and or on the level of exploitation of the plant. So how many individuals are exploiting the plant, there's this density-dependent response in them as well, which makes the problem ecological in nature. Notice how simple the ecology is. So in each generation, a fixed number of eggs make it to mature adults. So this is just a device to make the whole thing easy and not have any population dynamics. We have a certain number of individuals coming in, they lay a bunch of eggs, and the same number of individuals come out. So you might think this is an environment with a fixed carrying capacity. And so in our model we said, what happens if we have 4 environments? And we say, okay, this environment can produce 50 individuals, this one no individuals, this 50, this no. So we'll have a set of 4 numbers. Then we say, if we're in a 2-dimensional signal space, so every plant is putting out 2 components. Like this plant puts out 0 of component 1 and 1 of component 2. This plant 1/3 of component 1, 2/3 of component 2, and so on. Then we say the relative ratio is what characterizes the plant type. And when we have evolution going on, then we allow these ratios to be perturbed. And so we start off with 100 artificial neural networks. We run them on this habitat. And the behavioral ecology has to do with animals deciding what niche to put their eggs in, and they do this in proportion to the perceptual response. So if they have a strong response to one niche and not the other, like a 1-0, they'll only put their eggs in that niche and not the other. And again, fitness is determined determined by the quality of the plants that are being exploited plus terms that reflect competition. So we do some simulations, and in these simulations, if the simulation output is 1, 1, 0, 0 for a particular individual, it means that individual lays half its eggs in niche 1, half in niche 2, none in niche 3, none in niche 4. So that's kind of the output. And so a 0, 0, 1, 0 would mean that the individual is a specialist in exploiting niche 3. And we can specify our environment by having these niche specifications of their relative productivity. So niche 2 and niche 4 are 0, 0. That means they're protected and any eggs laid in there don't develop. And this is just the different ways to skin a cat. Here we let the fittest individuals produce 2 young and the least fit 0 young. And then we mutate the weights in our individuals, and if it's plants that are evolving, we mutate their signals as we go from each generation to the next. And we simulate for 100 generations and we get outputs like this. So when we start out, because the weights are chosen at random, it turns out that for these artificial neural nets, if you choose the weights at random in a particular way, everybody's just a generalist. Then after a certain number of generations, you get what I call a discerning specialist. Now, if this is habitat quality, you want to choose habitats 2 and 3, and not 1 and 4. So these individuals, within 180 generations, the population has come on its discerning generalists. And this goes on for quite a long time. And as you can see, eventually we get to the situation where we have 2 specialists on 1 and 3 existing to some extent with discerning generalists, but this goes in and out, and you get an invasion of a discerning generalist. This happens for a certain amount of time. So discerning generalists are not very stable, but they keep coming in and disappearing. If the habitat quality is more complex, so 30-20, 30-20, then we start out where everybody is a generalist. We can run it for a long time and we get generalists here are dominating the specialists. So for example, 1, 1, 1 is generalist. Here we have a couple of specialists. And so it's a complex picture, and then all of a sudden we can get a switch. So they're obviously sort of bowls of attraction that are quite shallow, and you can jump to different situations of stable configurations in the kind of guilds that would support. So the conclusion was that the emergence of phenotypes depends on the perceptual task. So for example, in the signal space, if that's component 1, that's component 2, and here you've got niche 1, niche 2, niche 3, and niche 4, and 1 and 2 are good and 3 or 4 are no good, then all you have to do in this space is draw a line like that and say everything above here I like, everything below here I don't like. So in a sense, in your brain, you have to evolve to be able to divide the space into 2. Here you have to evolve to divide the space into 3 bits. These extremes are good, and so you're much more likely to get specialists in this situation than a specialist in this situation. In fact, you're unlikely to get a specialist here, but you will get here. And then the hardest is when the edible and inedible plants are organized so that you have to divide the space up into 4 different components if you want to be able to recognize good versus bad. And in that situation, very often an individual will evolve to just accept things in here and another individual to accept things in here. So here's where you'll get specialists arising. So it depends on how individuals are configured in that space. And from an ecological point of view and on a slightly more mathy point of view, it turns out that the guilds form these so-called ideal free distributions in their fitness, and mathematically they also what's called locally adaptive Nash equilibria. They become more resilient to mutations as time progresses. Now in all of this the plants were static. If we allow the plants to evolve, we set up a model where we had evolving artificial neural networks with cloning and mutations in a heterogeneous plant space of varying quality, and competition also affecting the fitness of individuals, but the plants themselves have evolving signals in a two-dimensional space, which means those signals that they put out, that ratio can now also co-evolve. And plants reduce their quality at a cost to their fitness. So that means plants that are protecting themselves are slightly less fit than plants that don't have to protect themselves. And then, of course, plants that are heavily exploited are going to be less fit than plants that are lightly exploited. So, if we have a look at the evolutionary phases that you can get, let me just explain these diagrams. Underneath here, you have your plants sit on this space over here. That's your signal space. So, for example, all the yellow plants, which in this particular representation, the green are undefended plants. They're the best quality. The yellow are intermediate, and the red are defended plants, so individuals can't really exploit them. When you start out, there's no really good discrimination, and as we have in situation A, this blue surface here represents the response. You can think of it a response of an individual. So this individual over here responds in this part of the space, you've got a 1, but responds much less in this part of the space as the surface goes down. These surfaces here represent means for the population, not individuals. So remember, the blue surface is the average individual's response. So what happens is that as you go through this process, the first thing is you initialize it, and you put your plants down with a little bit of variation in different places, and your average individual is not responding particularly to anything, except that after you run it for a little bit of time, you can see that the average individual is starting to avoid the red plants in the middle. Then as you go to B, the most edible plants become mimics. So you can see the red is evolving towards being green. The average individual is recognizing the green and beyond where there's nothing, but as it comes into the green, it has to try and avoid the red, so the response dips there. And then the yellow is just put in to make it a little more complicated for the organisms to exploit. And then what happens is you get a red queen chase, in which the green runs around chasing the red, and the red runs around and eventually gets trapped in the corner. So this is, you can think of, the constraints on the kind of signal you can put out, so you eventually get trapped by your constraints in the signal space. And so what happens is when the reds get trapped in the corner and the greens become complete mimics of them, then the average individual can't discriminate anymore. And so they're not discriminating. So you lose, individuals now lose the ability to discriminate between mimics and the models of the mimics. So models in the mimic sense, not in the model I'm building sense. And so you continue with this process, and just through some random drift, these drift apart, and as soon as they drift apart, you can see here that individuals are trying to hone in on trying to identify the greens in the corner. But what happens is that the greens actually move through the reds, and in G, they move out to the middle again, and in H, the whole process starts again. And so what I've described to you is actually running our model, and seeing what happens. So this is a lot of fun, but an enormous amount of work. So this is basically, 2/3 of a PhD thesis doing all this stuff. And so all of the phases I described for you, you'll see evolve. And what happens is that this is a cyclic process. And so it cycles around and no equilibrium occurs. Now you can make this more sophisticated by putting in sexual reproduction and have genes both for perceptual and mate choice and plant. And so we did all this, and it became rather complicated. And this is a story in of itself. And what we found out is that resource preference for mating in a particular habitats invade because accumulated deleterious mutants not expressed when individuals mate assortatively. So what's happening is that this system is rather interesting because deleterious mutants can accumulate as long as they're masked in terms of the behavior, and they are masked in terms of the choice behavior, where individuals are choosing different habitats, but when hybrids form, that masking is lost. And so this is a way in which the hybrids are less fit, and this is not what we put into model, this is what emerged from the model. And as I said, I don't want to go into the story too much, but you can have a look at the publication, but the point is that it would be nice if I could build these models in a couple of months instead of a couple of years. So my contention is that you use MATLAB, R, etc., it takes a couple of years. And that what we're trying to do is move towards a place where this can be done far more easily in a couple of months. So what I described there was the beginnings of what I call the field of computational population biology. In computational population biology, you have individuals with an inner and outer world. In this case, the inner world was the brain processing, and the outer world was the resource heterogeneity. In more complex systems, the inner world, you can monitor things like the hunger state, the thirst state, physiology, all sorts of things. And in the outer world, you can make that more complex. You can have multi-species outer worlds and so on. You can see that if you want to build models where you ask questions involving the link between the inner and outer world, it gets rather complicated. And so you have these hierarchical structured formulations where the inner world can change at faster time scales than the outer world. So you have to deal with that. You have to then integrate analysis across your various scales, try and understand what's going on. And you may have formulations where you mix differential equations. Let's say you have differential equations modeling physiology or difference equations modeling the inner world of an individual, and then your outer world may be an agent-based model. So you're mixing these paradigms for modeling and scheduling. And so, of course, you can do this all with sophisticated computer programs, but you have to be sort of a coding geek to be able to do this. And that's what the dummies refers to. The rest of us who are not coding geeks, what do? So we have to go get somebody who can help us, or use this platform we're developing. So, as I said, we have an agent who has this inner and outer world, and we're putting this into a big computational platform. So if we have a look at the way modeling is currently set up, mostly in the boundary between the outer world and the inner world, our individuals have a particular age, sex, life stage, biomass, and we can characterize the basic demography, so to speak, of the population in this boundary. Then we can go to the outer world where we consider abiotic drivers, conspecifics, predators, blah, blah, blah. One of the things that I'm very interested in is pathogens and diseases, and it's becoming more and more evident that this is very important. Then in the inner world, as I mentioned, you have physiological and immunological states, and so we can either use lumped dynamical systems models, or we can use agent-based models. But if we want to go the full spectrum and mix everything together, this is what I'm calling computational population modeling. And the fact that I call it that is just to get people thinking that we're dealing with something more complex. And although this is a mixture of things that we know well, it's the mixture itself that's getting rather complicated. So what are the elements of a true computational population biology framework? The one is that we need to address real problems on real landscapes, and we do that now because we're collecting more and more information that has in our GIS databases, and we have a way to simulate individuals moving over real landscapes instead of just these toy ones that I've been talking about. We need better growth models that take into account how individuals take in resources, process them, turn them into growth, and turn them into reproduction. We have to pay greater attention in our demography to the fact that as individuals starve, they become more susceptible to diseases, parasitism, and so on in other parts of the ecological system. So we do this by having both demography and epidemics. And then we have these individual agents who have internal states. We have ways of specifying how individuals interact through connectance topologies. So this is, in particular in disease models, we're very aware of how individuals may transmit diseases through the individuals they socially interact with. We have perceptual modules like the artificial neural network model that I talked about, and these can be trained to, or can be tuned to compute certain decisions on what to exploit. In my particular case, I'm also very interested in using these modules in movement ecology and animals deciding where to move. So we have these different modules, movement and other action modules. So where to move, how to behave, what mates to choose, and then in the reproduction modules, based on mate choice, we have things that in the model that will control mate choice, litter sizes, and also we have to think about, are we going to have agents just making clones of themselves, or are we going to bring in sexual aspects and look at genetics and exchange of genes if we want to ask questions in evolutionary ecology. And then we have to have a good simulation engine, and I've talked about using MATLAB, and what I'm going to talk about now is using NOVA. But one of the things is that you get a pile of data, and you've got to know how to take this data and look for patterns. And one of the things I'm interested in is in looking at patterns of movement, and we've developed this tool called T-Loco, so some of those of you who work in movement ecology will know about that package, but visualization tools and ways to look at data and patterns is very important. So the thing is to describe the characteristics of a good computational platform. And this is, as I said, Richard started out with a particular platform that we've been developing. It's got a bunch of features in here that allow you to set up models using icons and graphically driven elements and produce code. You don't code this up. The computer codes this up based on what you're building here. And I'll talk a little bit about those elements. I'm not going to talk about too much about this. But I'm going to talk a little bit about the NOVA concept. So the idea behind NOVA is to promote collaborative community of builders. And the reason for this particular motif is here we have a tiger in a cage. And so it's what I call, building these models, I call riding the tiger. And if you want to ride a tiger, it's very, very dangerous. And so we've got to let this tiger out of the cage and ride it without falling off and getting eaten in the process. So NOVA is a highly expressive visual language which will help us set new standards of information exchange. It promotes process thinking and will thus lead to a better trained generation of applied scientists. And we're already using certain limited aspects of NOVA to go to the school environment and get kids playing around with web apps based on building NOVA. And in particular, the last year or two, we've been doing this in epidemiology. And it allows for rapid prototyping and accurate coding. And the reason why it's accurate coding is because you're actually not coding most of it yourself. You're using chips and things that I'll talk about. Base modeling practice, so this will promote base modeling practices and reproductive science. So I can see I'm running behind in time. So I'm going to go fairly fast through part of this and then get to a certain point. So NOVA is supposed to be easy entry, and but ultimately we want to have it spectacular power so that you'll develop NOVA on your desktop, and then once you've compiled it and everything, you'll be able to run it in a cluster and then import the data back to your desktop to work with the simulation. So there's some general principles that relate to modularity, scalability, interchangeability, specifiability, comparability, and interpretability, and applicability. So sorry for that, but I want to get to the more interesting things. So NOVA's got to do two things. It's got to incorporate the system dynamics or stock flow paradigm, as well as the spatial cellular automata paradigm, of which the game of life is one example. But instead of just looking at changing the colors of cells, you can actually move agents over landscapes, as in this wolf-sheep vegetation sort of toy. And it has these primitive components, much as you see in things like Stella and Vensim, and these are all generated graphically. But what you do is, after you build something that looks familiar to people who use, say, Stella or Berkeley Madonna, you turn it into a capsule by giving it these pins. And once you give it these pins, then you can drag it out in a new environment, and you automatically get an element that is an instantiation of this calculation, but can be used now at this new level of modeling. And so these pins also can be given their own clock chip. So you can open them up and you can set parameters. So for example, this pin, in one tick of the big clock that's running the model at the higher level, you can solve a differential equation at the lower level. And then you've got these different capsules, and one way of containing all this is to develop what are called these aggregators. So one is to organize cells into a cell matrix. The other is to organize agents into an agent vector. And an agent vector specifies how the capsule works dynamically, what its location is in space, and where it is in time. And so you can look at its spatial position, its temporal dynamics. So you first create the agent, and then, as I showed you before, you can go to a higher level there, and you can drag this agent onto this new canvas and get this sort of encapsulated as a clock chip. So everything that's going on in that chip at the higher level is built here at this lower level. Here we have an aggregator called a sim world, and in a sim world, you're connecting what happens in a cell matrix and what happens in an agent vector. You can specify 100 agents on a 30 by 30 world. You drag the agents onto this icon, you drag the cell onto this icon, and then you've got everything coded up on how the agents are going to work on this cell world, and you can move them around. Here we have another kind of aggregator, which is networks for disease problems. So for example, instead of running things on an array, we can run them on a network, and you can run a disease model on this network, and you can plot the output. So this is just sort of giving you a quick overview of some of the things. So you have these code chips, so that if I want to build a model and I want a certain functionality, as long as there's a bunch of code chips out there, I can pull in the code chips I need. So I don't need to write this code, which happens to be an extension of JavaScript. And so they're easy to implement, import and export. We also have what's called these Java plugins, which help in visualization. For example, you want to make a histogram. You go to the plugin menu, you choose the histogram, you drag it out. You select it, you drag it out, and then you set the specs of the histogram. and then you connect up the data you want to come into this, and this is all done, and it's menu driven, so you don't have to know how to code in JavaScript to do all this stuff. Oops. So we did that. And for example, in the models that I was talking about, we had these artificial neural networks, which are multi-layered perceptrons. So we have these tools as well, which can be used. Sorry. So there we're running a multi-layered perceptron, so you can just use that. You don't have to code it up. You just place it onto the canvas and you take the relevant information. And so once you've done all this, you get your NovaScript program, which is really an extension of JavaScript. You'll create the program, which is a JavaScript execution, and you can just run it on a Java real-time run environment. You don't have to know any of this. And so you could also build these models from scratch. So if you knew JavaScript or NovaScript, you could write it this way, or you could just build it like that. So that's the real difference, and that's why you don't need to be a coder to do this. So what I'm interested in is these genetic algorithms. So getting back to these genetic algorithms, you have an individual, you have a patch equation, you have an individual equation here. This individual gains biomass. It has rules on where X, Y, where it's going to move. It has a little module here on where to go. If you don't like this module on where to go, there may be another module that somebody else developed on how an individual chooses where to go. You can run your model, then you can replace this just with a drag and drop, and you've got a new model that tells you, okay, the animal's making decisions in different ways. And so you initialize the sequence. The where to go examines what's going on and says, should I move or stay? If I stay, I can extract resources locally. If I move, which is the best direction for me to move in, and where should I go? And when you do that, you come to the level of, you've got the sim world. Each agent here is dragged out. The cells onto here, the cells onto here, the agents onto here, and then you get this visual graphic. We actually, then you can run that, and at the end of each time period, you can see who collected the most resources. Then you can go back, but you can have an evolutionary level where you run this many times and you allow the parameters of each individual that was successful and reproduced to have new parameters and to have those mutate. And so basically you get a genetic algorithm that's going to run. This is kind of what a run would look like. So finally, the success of computational population biology depends on having an active research community that adopts an open module approach in developing tools and can share the different code chips that people develop. So you'll have people who with coding skills developing code chips, and then people who don't have the coding skills still being able to build sophisticated models, provided suitable code chips are available. And if you need a code chip to do something and you don't have it available, then you persuade somebody who's not a coding dummy to do it for you. And so I am interested in trying to build a community of users. It's a difficult process to do this. One of the things we have to do is think about protocols for naming modules and presenting model structure. So instead of presenting your model as a bunch of code or the description of equations or coding statements, you actually have these graphical pictures. And you can look at them at different levels and you can see how things plug together and how the system works. And then of course we need to know how to validate modules and the models that they built from. And this has and one of the ways of moving forward is to promote comparative studies so that we can say, well, what would happen if we use this kind of movement module versus that kind of movement module, where everything else remains the same. And in this modeling platform, it'll be very clear that everything else has remained the same because we'll have these graphical representations of what the models look like. And so we need to pay more attention to developing software tools for education and for analyzing data. And most importantly, and I think this is very important, is that journals need to set new standards for accepting modeling studies in terms of replication of simulations and presenting model structure and code. And I think these kinds of representations and communication of model structure and code is going to be more easily done in something like a Nova platform. And so I know I've gone over, but this work has greatly benefited from numerous students, as all professors here, almost all, how good they are depends on how good of students they got. And Nova architecture is due to the genius of Richard Salter, not to me.